we're going to talk a little bit about moonlighting. Now, this process over the last 10 years it's really become to our attention that this is way more common than what we previously thought. There are multiple ways for proteins to take on extra jobs, right? You can do it by changing location. You can do it by changing shape. You can do it by changing the interaction you have, right? Enolase, one that we're going to talk about today, changes its oligomeric state, changes its shape, and changes its location all at the same time. So, like I said, this process occurs in every organism we've looked in, all the way from humans until mycoplasma. If we track enolase, which is the penultimate step in glycolysis, at least that's what we think its primary function is. The more I think about it, the more I would argue that its moonlighting function is its primary function, but I'm not going to get into that today. Right? But enolase is highly conserved from humans to mycoplasmas in the glycolytic cycle. It's also highly conserved from humans to mycoplasmas on the surface of the cell. Okay? Now, John Sanford, our graduate student that spoke right before me, his work has shown that these particular moonlighting proteins are hexosylated on the surface of SYNT3A. CTP synthase, elongation factor, EFTU, lactate dehydrogenase. These are standard, well-known moonlighting proteins, right? Now, in pulmonis and arthritis, we showed this more conclusively as a rhamnophospholipid. In Mycoplasma mycoides, GM12 strain, I have evidence that it's a di-rhamnophospholipid that links these proteins into the membrane, okay? So if we look at where the PLD cleavage site is, this is where we would expect it to cleave. And so that's the enzyme we utilize, phospholipase D. Like this is that slide where I was talking about the shape of the, what could manifest into that. This is what I was talking about. This is the mycoides subspecies GM12, the di-rhamnophospholipid linker that links these moonlighting proteins to the outer membrane surface. Okay, we have shown this fairly conclusively in arthritis and pulmonis. We have strong evidence for this in mycoides, and we have hypothesized that the enzyme phospholipase D would cleave this at this site right here. So we ran SDS page gels of, this is just equivalent amounts of SYNT3A supernatant. The cells were spun down after the enzyme reaction. We acted one with phospholipase D. We reacted the other with heat kill phospholipase D. Now, SYNT3A cells are a little bit sensitive in how you handle them. And this, we finally ended up doing this experiment with some glycerol, about 5% glycerol, just to stabilize the cells enough so we didn't get a whole bunch of autolysis, basically. And so this is where DNA K is. This is enolase, and this is EFTU. Now, if we excise one of these bands and take a look by GCMS, and in this particular case, we're looking at this band right here, this enolase band, and analyze it by GC, we see a couple of things. This is the mannose in A. I can tell what it is basically by the mass spec signature. Okay, all of these are unique for the molecule. If you combine that with the time and standards, you can get fairly accurate as to what it is. This is PQR's mannose. This one is mannose phosphate. This is glucose from the hexosylation system that John talked about. And this is our lipid linker. Now, GC has a very unique peculiarity in that lipids show up about 10 times stronger than everything else. So this lipid, I mean, if it was a sugar, would be about here where this noise is, right? Concentration-wise or quantity-wise. You can tell it's a lipid by the breaks of 14 all through the mass spec. Okay. So what that means is in synth3A, this is a dimannophospholipid linker. Okay, not a diramnose, a dimannophospholipid linker. This is also what I found in synth1. So rhamnose synthesis is knocked out in synth1. Okay, so now if we look at the just synth3A cells using flow cytometry and antibodies, in this case, we've got one that was generated against Staph aureus, and we've got one that was generated against human. And both of these are against enolase, and they are certainly on the surface of the cell. If we look at human DNA-K, that's what these antibodies were generated against. These are all against synth, obviously. Or human EFTU, we can once again see that we get a really strong reaction. So mycoplasma, the moonlighting system, is active in synth3A. All right. At a minimum, 40% of the proteome has a secondary function. In these minimal genome organisms, I would propose that that number is a lot closer to 75% of the proteome. In the case of synth3A, that would give you 300 extra functions. Questions of essentiality, they become way more complex. Is it the primary, the secondary? Which one is it? When we found these things, we arbitrarily assigned the primary function to the one we found first. And very little biochemistry has been done on any of these enzymes. But in mycoplasma, I can think of one or two cases where proper biochemistry has been done on the enzymatic pathways. And AI and homology is just going to take off and follow E. coli down a path and settle us down a path because that's what the computer is going to tell them. This system is far more complex than that. Right? Over the next 6 months to a year, I'm going to spend my time trying to understand the moonlighting system and pathway in mycoplasmas using synth, the tools I have in synth. So, but it also opens up unique possibilities, right? So how far can we actually shrink the genome if every member of that pool does 3 or 4 functions? Can we really make a cell with 100 genes? I don't know the answer to that question, but I hope I stick around long enough to find out. Thank y'all for being your attention. And thanks, John, for hosting this once again. We sure appreciate it. I'll be happy to take any questions.
Thank you very much, James. I struggle with, so you give evidence of enolase, DNA-K, EFTU, likely. So with enolase, we know that it has its glycolytic function, and we also know it's a member of the RNA degradosome.
Hmm?
With DNA-A, I mean, with DNA-K and with EFTU, do you have any sense of what the other functions might be?
John, I truly think, at least in enolase case, because if you look, you find it on the surface of human cells, just like you find it on the surface of mycoplasma cells. I think that function is just as essential. Maybe it's stabilizing. Maybe it's stabilizing the membrane. Maybe it's interact. If you look at the online databases, they tell you it's plasminogen binding, and I don't really buy that.
But you've also got the RNA degradosome.
True.
Which is clearly a second essential function.
Yes, that's just one with a tertiary function.
And I got no problem with any of that, but how do we know that just because the protein, some fraction of the protein makes it to the surface, that it may not have a function?
Well, if you look at like examples like Mycoplasma arthritis or Mycoplasma arginini, they don't have a glycolytic cycle at all. They don't need one. They are arginine used. They utilize arginine. They maintain at a high homology enolase, and it is found on the surface of Arthritis and arginine.
But they also have to have an RNA degradosome.
Absolutely.
So there's no choice in that one.
True. I'm just saying that function can't be possible to be that widespread, because you find it on the outside of human cells too. And it was.
Go ahead, please.
You too, John. That was about all my thought anyway.
So one of the rationales for building our minimal cell was to see if we could come up with a list of all the, not the genes necessary for life, but the tasks that a cell has to do to be alive. For instance, a cell has to have lysine. So you can have one transporter protein, or you can have 30 proteins that make lysine. Either equally good. But the cell has this list of things it has to do. And so it seems like you're suggesting that even this simplest of all cells is doing a whole lot more things than we as biologists are aware of.
That's exactly what I'm suggesting.
I like it. It's until we can start. So, part of me says, look at the AlphaFold predicts what proteins do. And so we tend to stop when we get a great prediction for one thing, but that secondary prediction may be equally important.
Yes.
Things that are 40 or 60% likely to be X. And at least that would give you a starting point to think about. We could use the free gene set to purify a protein from, an in vitro system or something, or make a bunch of it in E. coli, and then think about doing biochemistry on this.
Absolutely. And that's what I'm going to try to do over the next 6 months to a year, is really use immunoprecipitation, since I have antibodies against these surface membrane fighting proteins. Pull out those 3 proteins. I've already shown the antibodies work. I think I can get them. And let's see if we can figure out where the site is. Is there a amino acid sequence specificity? Is there some commonality that we can pull out and predict where these things are going to occur? That's the goal. If I've got 3 of them, I've got a shot at it. And I've already got 3 antibodies that are working pretty good, and I'm pretty sure IP will work. So